Hello, welcome to St. Charles High School's College Polo Day. As you can see from the red hawk on my chest or the diploma over my shoulder, I went to SEMO, or Southeast Missouri State University. Had a fantastic time there. I learned a lot, had a lot of great experiences. Any questions, you know where to find me. I'd love to talk about SEMO. But what's more important to us that live here and now is our next unit, which is basic biochemistry. It's the chemistry of all living things. We'll talk about why this is important to biology, really the foundational things that we're going to move out of with the graphene and the metric system experimental design and get into more actual content as far as the course is concerned. So let's begin. Atoms. Chemistry is the study of atoms and their interactions. So not only the stuff that makes up matter, but also how that stuff interacts with one another. We'll learn that they interact because of interactions with their electrons. You can see electrons are one of these three component parts. Protons and neutrons are in the middle. Electrons go around. They can be varying distances away from that nucleus depending on how much energy they possess. So protons have a positive charge. You see the P plus here. Neutrons are neutral, as in they have no charge. Sometimes you see those denoted as N0. And then electrons are negative, or E minus. They travel around. So why is this important to biology? Well, chemistry helps explain the structure and function of all cell processes at that molecular level. So really drilling down deep to what actually happens with these interactions, not just at the cellular level, but even deeper than that with the organelles in between and the individual compounds, which we'll learn about in a moment, that exist that make up these things. And that's really where we're leading to. These atoms interact with one another because they swap electrons. So remember from last slide, you've got positives and negatives. So if you lose an electron, or if you lose a negative, all of a sudden you have more protons or more positives than you do electrons or negatives, so you have an overall positive charge. If you gain an electron, now you've got more negatives than you do protons or positives, so you have an overall negative charge. And then these opposing charges, much like my wife and I, who are total opposites, attract. Maybe you'd learned about this before in middle school or in elementary school with magnets. Positive negative pole or north and south pole will actually work and attract towards one each other. Like conversely, the like charges, two negatives or two positives, will repel. Very good. So here's three examples. This first atom is carbon. How do I know that? We'll get to the periodic table here in a moment, but 6 protons means it's carbon. The neutrons are again neutral, so they don't really affect the electrical charge of the atom or the element. But 6 protons and 6 electrons, those 6 cancel out these 6, and sure enough, you have a neutral atom. This next one here with 5 protons, well, that's boron. Again, you can see that right here on the periodic table on the back of your blue page. That boron has 5 protons, but this particular one has 6 electrons. So there's one more negative than there are positives. Those first 5 work together to cancel each other out, but now all of a sudden you've got an extra electron out there on the end, this little guy. So that makes the overall charge negative. The last one, this atom, is positively charged. We're back to carbon. 5 protons, 5 neutrons, but now we have 5 electrons. Sorry, 6, 6, and 5. I misspoke. So you have one more positive than you do negative. Sure enough, this atom is positively charged. So I talked about compounds earlier. Compounds are made of 2 or more bonded elements. So there's different type of compounds. We'll get to those in a second. They depend on the type of bond. Here's an example. We've all heard of water. We had that in a Penny Lab we did 2 weeks ago. But hydrogen plus oxygen equals water. Now we'll talk a little bit later about the number of electrons in the H2O like you can see in this example. Here's another one. Carbon plus oxygen plus hydrogen is a carbohydrate. Think carbo, hydrogen, and oxygen. We learned up here make water. Hydra, hydrate, so carbohydrate. These compounds make up the cells that living things are made of. And that's really where we're going to get to. Not so much today, but we're going to talk about this basic biochem stuff now so we could really lead into our 4 classes of organic macromolecules. There are carbs, proteins, lipids, and nucleic acids. And we'll get to those in due time. Here's another example of a carbohydrate. There's C, I think this is C6, H12O6. The most likely is glucose. I'd have to look closer. So isotopes. Earlier I said that we'll talk about neutrons more in a moment. The time is now. So isotopes are forms of an element with different numbers of neutrons. This affects the atomic mass, but not the chemical identity. Remember a couple slides ago when we said, hey, this is an example of carbon that's neutral, this is an example of carbon that's charged. I knew both examples are carbon, and now you do too because they have 6 protons. The protons are what give something its chemical identity. The number of neutrons is going to lead to its mass. You can have greater or fewer neutrons, and it doesn't change what element you have because the proton number doesn't change, but it does change the mass. Remember, the neutrons are neutral, so they do not affect the charge, therefore do not affect the chemical identity. Good way to remember this is if you have identical twins and maybe one weighs more than the other. They're alike in every way except for their mass. I guess that's a very simplistic or rudimentary example. Here's an example here of isotopes of hydrogen. You got this one with one proton, no neutrons. All of a sudden you add an extra neutron. Well, it's still hydrogen. There's still one proton, but you've doubled the mass. Remember earlier I said that they have approximately similar sizes, so they each weigh 1 AMU or atomic mass unit. Here's another example, only this one's carbon. And we're going to talk about carbon-14 more next semester, but carbon-12 is kind of traditional carbon. You know, standard carbon is the majority of the isotope that's out there, but there's a set ratio, carbon-14. It's got 6 protons still. Again, it's still carbon, but all of a sudden we've added 8 neutrons, so it weighs extra compared to normal carbon here. So let's talk about energy levels. I said earlier that electrons can be varying distances from the nucleus based on how much energy they have. So the bigger your atom is in mass terms, the more energy it's going to have, and the electrons are going to be spread out. Again, this is very basic. There's far more depth than this. We're just trying to keep it simple to establish that foundation that we're going to build from, okay? So each row on this periodic table represents one energy level. 1 energy level, 2, 3, 4 energy levels, and so forth. The number of columns are how many valence electron. Here's a new word. Valence electrons are present. Valence is a fancy term for, hey, what's on the outermost shell. Each shell can only hold a certain amount. The first shell or first energy level holds 2. The next one holds 8, 8, and so forth, right? So, however, are in the outermost are what's going to actually interact or react with other atoms or other elements. Everything wants to have an octet. An octet, think oct means 8. So an octet rule is what we call a stable isotope, or a stable version of that particular atom or element. So anything with more than or less than 8, it's going to want to react with another element to work out some sort of deal. Maybe they're going to share electrons, or maybe they'll just outright transfer or steal one from another one. And that's where we're going to get into our different types of compounds, because they're going to want to react differently. So the number of valence electrons is important because it's going to determine how or in which manner is most likely for an element to react. It gets far more complicated after the third row. We're going to kind of leave off at calcium, or element number 20. It's about as deep into the fourth row as we're going to go. We call those advanced chem problems or honors chem problems. Again, we're just setting you up for our organic macromolecule unit. You'll get more in depth with chemistry next year in honors or advanced chem. So one type is called a covalent bond. All atoms want stability, like I said before. We call this the octet rule. Again, I just said that on the last slide. They sometimes share electrons. This is called a covalent bond. You can see in the example here, you've got water. All of a sudden, here's an oxygen. So look, they're sharing here. Here's carbon dioxide. Here's oxygen, here's carbon. So again, carbon dioxide. Covalent bonds, you're going to see those a lot in your nonmetals. Over here on the right side of the table, past what's known as the staircase. So you see a lot of carbons, oxygens, a lot of the things over here. When two nonmetals bond, they tend to share those electrons on their valence shell. Notice the ones in the middle here. Sorry, I tried to point with my finger. You can't see that. Notice the ones in the middle, all right? They're not really reacting. It's the ones in the outermost shell. Same thing here. The first row holds 2. The outermost ones are what react. Again, it's those covalent. Again, notice how the center of each one is charged. That's the protons. The electrons all have negative charges. So when things share electrons, we call that covalent. All right? An ion is a charged atom. A cation is positive, and an anion is negative. An old school TV, not a flat screen, might be called a, you see a cathode ray tube. You could really, if you use cathode ray tubes, you can really see the positive and negative nature of some of these, but that's an experiment for another day. Their opposing charges attract, like we said before, and this creates an ionic bond. So this happens a lot of times when you have a metal and a non-metal. For example, sodium chloride. You see that right here. Sodium is looking to get rid of this one, so it has 8 in its outermost shell, or octet rule. And chlorine's looking to add. Right now it has 7, so it's looking to steal one. So they work out a deal where Sodium is willing to give one away. Chlorine is willing to take one on. So all of a sudden you have sodium chloride, or what's known as table salt. That makes sodium a cation, makes chlorine an anion, but now they're connected. They're now an ionic compound or a crystal of some sort, as opposed to the covalent compounds on the last slide, which are molecules. So here's a nice little graphic to sum it up. Here you've got a covalent bond where these things share electrons. And here's an actual transfer of electron outright, or a stolen electron in a ionic or anionic bond. The last we're going to talk about are hydrogen bonds. Now these are going to come up again and again, especially when we talk about DNA replication, transcription, translation, things like that. Hydrogen bonds, they're very convenient. They're very weak, but at the same time, they hold things together when need be. It's not like Velcro shoes that, you know, maybe your dad might wear around or a grandpa. I know my dad just can't get enough of the Velcro shoes. It's whatever, all right? So hydrogen bonds, some molecules have actual charged poles where they form weak bonds, just like we saw in the Penny lab with that surface tension. Here, you've got the larger mass of the oxygen atom actually pulls the electrons toward it. That creates an area of partial negative and partial positive charge. We call that sigma partial, and sigma partial is sigma neg and sigma pos basically. So they haven't completely stolen. It's still a covalent bond, but the electrons stay closer to the oxygen because it has many more atomic mass units than is these tiny hydrogens. So because of that, here's an example of water, H2O. All the partial negative areas kind of link up with the partial positive areas. And you see this a lot with water. That's why it kind of has that capillary action and that meniscus we see whenever we're measuring the volume in a graduated cylinder like we did last week, or why water tends to bead up whenever you spill it on a tabletop. Okay? And that's what led to our ability in the Penny lab. So which elements do we need? Again, this is biology. These are the big three that make up the most mass of living cells. There's extra things that are in there. We're going to talk a lot about iron or hemoglobin when we get into proteins, especially quaternary proteins, that not only fold in more than one level, but also form with other proteins to make super proteins. And that's, where hemoglobin comes in, and iron or Fe, those ions, have a lot to do with that. So we're going to get into the periodic table and how to read it in far more detail starting next class. We'll talk about Lewis dot diagrams. We'll talk about Bohr models as a way just to identify where these electrons are and what's most likely to join with what. And that's, again, going to lead us into our organic macromolecule unit where you're talking about carbs, proteins, lipids, and nucleic acids. All these are just compounds. Remember, that's more than one atom bonded together chemically that form in different ways and therefore can have different reactions with one another. Thanks.